champions, how are you all? I hope everyone who is watching this video is doing absolutely happy, is doing good and everything nice. And I also hope that everyone is high in Josh, right? Please not be worried that exams are nearing, we haven't studied anything. Bacha Pati, we are here for you guys, right? And also you are giving your best, I'm pretty sure of that, which is why you are here watching this particular video. So if you think that you have not given your best until date, it's okay. From today onwards, start giving your best, and whenever you give something, okay, from your end, you will be seeing the results as well. So today we are going to start talking about the model paper 1 solutions. Are you ready? If you're ready, just let me know that you are ready in the comment section, and also do let me know whether this particular session was useful for you or not, because whenever I see this comments and all, I get motivated. Just like how you need motivation, I also need motivation, right? So you have to tell me about the negative feedback, tell me about the positive things, everything together. So let's get started, my dear champions. Okay. The first question, part A, my dear children, part A consists of 1 marker questions. So we are going to solve certain MCQs. Chalo. The first question states, statement 1, apomixis is the production of seeds from unfertilized ovules. Is it? Is it? From unfertilized ovules, how can you get the seeds? So usually what happens is that ovule, inside the ovule there is an egg cell. When this egg cell meets with the male gamete, that's when we say that the fertilization has happened, and this gives rise to what? Zygote. When fertilization has happened, now we will call this particular ovule as what? A seed. Now, according to the statement, it is said that apomixis is the production of seeds from unfertilized ovule. From unfertilized, if the ovule is basically the egg is not fertilized, can you get zygote? No. If you do not get zygote, can you get seed? No. That's what we usually think, but it's not like that. So apomixis is actually a procedure where you do not need the male gamete to come and fertilize. Basically what happens, this unfertilized ovule itself will now be turning up into a seed, and it's not found in all the plants, my dear children. It's found in very limited number of plants, and this is a procedure which is already there like that. So there is no need of fertilization, still the ovule will become seed, and this process is known as apomixis. So this is entirely correct. Statement 1 is true. What about statement 2? Embryos produced from apomictic seeds are not generally identical to the parent plant. So suppose the parent plant, the female parent was giving this particular thing, right? Ovule. Ovule is there in the female parent. Now, I am saying basically the egg cell which is there, it will only form the zygote without fertilization. It will only turn into an embryo, everything. So basically, didn't I start from a cell that is there in the female parent? I started from a cell which is there in the female parent. So it will have all the features of the female parent. Right. When the male parent and the female parent's genetic material comes together, that's when you get a new type of progeny which has the genetic material of both the parents. But as of now, when you talk about this, okay, here only the female parent is involved, only the female parent's egg is involved, there is no fusion of gametes. So do you think the progeny, the embryo which will be formed, it will be totally different from the parent? No, my dear children, it will be entirely similar to the parent that is there. So embryos produced from apomictic seeds are not generally identical to the parent plant, but be wrong. They are identical to the parent plant. Why? Because only one parent is involved. And can I say that's asexual mode of reproduction? It's more kind of asexual mode of reproduction where there is no fusion of gametes. And what happens in asexual mode of reproduction? The progenies which are produced are more or less like their parents only. That is why we say they are producing clones. Similarly, here also they are producing clones. So I'll say the statement is false, but statement 1 is true. So statement 1 is true, statement 2 is false. This becomes your answer for the first question. Now, going ahead with the second question, choose the correct option from the table given below for the formation and dissolution of the labeled part in the given diagram. So the labeled part is what? Zona pellucida. So what happens, my dear children? So when egg cell is formed in the females, okay, in the human females, they are stuck in a phase, okay. They have not yet become the ovum. They are known as secondary oocyte because they have not completed the meiotic division. They are stuck somewhere. They are stuck somewhere. They have not completed the division. When do they complete the division? When fertilization and everything happens. At that point of time, they complete the division, and that's when the secondary oocyte which is there, it will divide again and give 2 cells. One is the second polar body, and the next one will be the ovum. So at this point of time, it is stuck. And when it is stuck in this stage, it produces an extra layer that is there. And this layer is known as zona pellucida. This layer is known as zona pellucida. Now what is zona pellucida has? Zona pellucida has certain proteins, my dear children. On these proteins, what will happen? The sperm which are there, they will come here and bind. They will come here and bind, and that's how fertilization everything happens. Now you are asked about choose the correct option from the table given below for the formation and dissolution. Who is responsible for the formation of zona pellucida? It is secondary oocyte. You know it is secondary oocyte. Now, when do you think this zona pellucida will be dissolved? It will be dissolved after fertilization, my dear children. So what happens? Initially this guy will come in, okay, will attack, and then when this attachment happens, there are so many enzymes up here, okay, in the acrosome of the sperm. This will be released, clearing some space up over here such that this particular sperm can get inside. Later what will happen, there will be certain changes in the zona pellucida. Okay, there will be, when this guy fuses with the secondary oocyte, there will be certain changes in the zona pellucida such that it does not allow the entry of any other sperms at all. And then after some time what will happen, slowly the zona pellucida layer that you see, it will dissolve. So it happens after fertilization, that is dissolution would be happening after fertilization, and it is found by a secondary oocyte, my dear children. So this becomes your answer. Cool? Works? Chalo. Next question. Out of the options given below, choose the correct stage for transfer of fallopian tube for successful IVF results. So here I am explaining you the MCQs, because MCQs are the place where you lose a lot of marks. About 5 marker, 3 marker, we won't be going in depth because we have already discussed in Brahmastra, but still I'll be telling you the answers, what all point you need to have. Okay, just for you to have a reminder, because I don't want this video to be very long, very boring for you guys. Cool? Just informative. That's the only purpose. Chalo. So, after the options given below, choose the correct stage for transfer to fallopian tube for successful IVF results. So, IVF means in vitro fertilization, my dear children. Now, for IVF, what do you need? Okay, which type of blastomeres embryo do you need? You cannot, suppose I am putting the embryos in the fallopian tube. Now suppose the embryos are of more than 16 blastomeres, that is 16 cells. Do you think they can grow over here? No, my dear children. First thing, in the fallopian tube, your embryo should not be growing very much. It should be moving down towards the uterus. So over here, when you're putting, you have to make sure that the embryo is only up to 8 blastomeres. Embryo is only having up to 8 blastomeres and not more than that, because slowly this embryo will divide and come down by itself. If you're keeping a whole big embryo over there, it will grow there and it will lead to something that is known as ectopic pregnancy. Okay, this is not something that we need. Ectopic pregnancy, something that will be dangerous both for the mother as well as the fetus. So up to 8 blastomeres you can put it in the fallopian tube. More than 8 blastomeres, if it is there, you can put it in the uterus directly. Okay, according to IVF, in vitro fertilization. Now the next question that we have, my dear children, 37.2% recombinant Drosophila progeny obtained in the T.H. Morgan's dihybrid cross experiment with the phenotypes red eye color normal body and white eye color miniature body is due to. So it is saying in basic sense, okay, so there is 37.2% recombination, that means new combination is being arising over here, where between the red eye normal body and the white eye miniature body organisms. Okay, now let me tell you, if you talk about recombination, 50% is the highest amount of recombination. When you say 50% recombination has happened, that is the highest amount of recombination that could happen. Now over here, when you are saying 37.2% recombinant Drosophila is there, that means the recombinant percentage is that much, is way too much. It's almost near 50. It's not 50, but it's towards 50, right? If I say that 1% recombination has happened, that means it is too less recombination. Now the question is, why does this recombination happen? Why does this much amount of recombination happen? Recombination will happen in less amount, in more amount when, my dear children, the genes are located very far from each other. That's when recombination will happen. What do you mean by recombination? First question. So basically, suppose these two genes are there. Now, I am going to make a new combination out of it, which would look something like this. So this part of the chromosome that you see have moved towards this side, okay, whereas this part has come towards this side, which is why the red line that you see has now gone here, whereas the other one plane is over here. This is a new combination of chromosome that I am getting. Here also I'm getting a new combination of chromosome. Initially this gene and the other gene was on the same chromosome, but now I'm getting a new combination. Why do you think higher recombination happens? Because the genes which are there, they are loosely linked. When they are far away from each other, what do you say? It is loosely linked, and also they are having higher distance between them. So can I say loosely linked and longer distance between the genes? This is the answer. I can say that. It's because they are loosely linked and longer distance is there between the genes. That is when the recombination rate is very much high. Suppose this guy was here. Suppose this guy was here, my dear children. So whatever you do over there, it will be like that. And suppose if you're cutting this portion and put it over here, both of them will be coming over here. Both of these genes would be coming over here. And here I won't have anything because they are very closely linked. That means recombination rate will be really less. So if you want more recombination, that means that it should be loosely linked and longer distances should be present in between the genes. Got it? Chalo, we have completed 4 questions. Yay, tap your back. Now, next question. The number of nucleotide pairs present in the DNA of the primary oocyte of a newborn human is. This question I basically made it wrong. Okay, somewhere in the Brahmastra I have to rectify it, but I am telling you right now. Okay, it's because I didn't read the question properly and I don't want you to do the same mistake again. The number of nucleotide pairs present in the DNA of the primary oocyte of a newborn in human. Now, I'll tell you my logic which I applied earlier is that basically whenever I talk about gametes, gametes have half the number of chromosome. They have half the number of chromosome. Okay, so for example, ovum, if I talk about ovum, it has half. Okay, now if I talk about sperm, it will be having half. What is half? Basically, the content will be 3.3 into 10 to the power 9 because the full content is 6.6 into 10 to the power 9. Now, if it is ovum, if it is sperm, that's when the amount of chromosome, basically amount of DNA will be half, because half and half will come together and make a full in the case of zygote. That is what usually happens. So, I use that logic since it is primary oocyte which is mentioned over here. So, primary oocyte is basically talking about the gamete of the females, right? But I had made one mistake. What is that mistake? When you are saying primary oocyte, my dear children, it has not underwent the whole meiosis cycle and divided into 2 to make it into half. For example, here also it is diploid. It is not haploid. It becomes haploid when it divides once in meiosis 1. But this poor primary oocyte is stuck in this stage. It is in the diploid stage only. Later you get the secondary oocyte. Secondary oocyte will again divide into 2, giving N and N. This is again meiosis 2, my dear children, giving ovum. So if it was ovum, then it would have been N, that is 3.3 to the power 9. But over here, when you're talking about primary oocyte, it has not yet completed its division. It's stuck in prophase 1. So can I say it is diploid only? That means it has full number of the same. The normal cells, how much number of chromosomes they have, or what content of DNA it has, the same here as well. So the answer will be 6.6 into 10 to the power 9, not 3.3 into 10 to the power 9. So if you're not aware about primary oocyte being like this, you would not be able to answer this. So when I went to the question once more, I was like, Jasmine did some mistake over there. So it's our duty to learn from our mistakes, which I politely apologize for all of you if you have made some confusions over there. That's my apologies. Okay, I didn't read the question carefully, but I don't want you to do the same mistake again. And from now on, you can just rely on me. It's not like whenever I have did a mistake, if at all I have rectified it. So please forgive me for that and just study what I'm saying. Okay. Now, always as a good student, you should be challenging me when you see questions like this. Okay, but no one either complained, so I also didn't get it, because when you are solving live, you do not think about it very much, right? So yes, okay, so I'm so sorry, but get your answers correct. Now, the factors that affect Hardy-Weinberg equilibrium are listed below. What do you mean by Hardy-Weinberg equilibrium? So, according to Hardy-Weinberg's law, what happens is that the allelic frequency of a population will be same generation after generation. That means my kids, everyone will be having the same allelic frequency. Okay, that is, for example, I have allele A. capital A in the population, there is small allele A also. So the frequency of capital allele and the frequency of small allele will be same. That is what is said in Hardy-Weinberg principle. Okay, but is it true? No, my dear children. For example, suppose an organism from that group from Africa came over here in India, and now you have a different allele altogether over here, or probably you have others that are having so many AAA alleles coming over. There are so many organisms coming like that. So basically you're disturbing the allelic frequency, right? So anything can change the allelic frequency of a population. It can be crossing over. Crossing over means when the recombination, everything happens, that can change the allelic frequency. Okay, or basically mutation, that can change the allelic frequency. Independent assortment, that can also change. Independent assortment as in when basically what happens is that, suppose this is a chromosome for capital R small r and this for capital Y small y. Now this chromosome can go with this chromosome in a gamete. In a gamete it can be round and green, or on the other hand this chromosome can go with this chromosome, that is it can be round and yellow also. So it can be either combination that can go. So obviously independent assortment, mutation, crossing over, genetic drift. What do you mean by genetic drift? For example, any sudden change in the population population alleles. For example, there is one area where there are green beetles, lot of green beetles up over here, okay, and in between them you have got red beetles. Okay, so white beetles and red beetles are not green, I make it white. So what happened in once, so over here if you see, the allelic frequency of the white ones were more than red ones, as you can see. One day one elephant came and stamped over here. What will happen at this point of time? All of them will die. But now what is a new thing that I am able to see? The new thing that I am able to see is that there are so many red, okay, and there is only one white. So again the allelic frequency has changed. So anything that is mentioned over here can be a reason. So I will say 1, 2, 3, 4, everything is correct. Can I say that? 1, 2, 3, 4, every option that you see over here is correct. All of them are the factors that affect the Hardy-Weinberg equilibrium. So you should be knowing about the factors: genetic drift, gene flow, mutation, crossing over. All these things are really natural selection. All these are the factors and you should be knowing about all of them. Cool. Next question. Interferons are most effective in making non-infected cells resistant against the spread of which of the following diseases in humans? So interferons, basically what do? Suppose one cell is affected. These cells are going to release something that is known as interferons, and these interferons that you see are going to alert other cell that, hey, there is some problem happening over here. Okay, so now the question is, which is that disease in which basically interferons play a major role? Which is the disease in which interferons play a major role? Now I'll tell you one big clue. So interferons are released when the cells are affected by virus. So viral infected cells are going to release interferons, and these interferons are going to act on them and say that, hey, there is an attack by what? The viruses, and you have to stay safe. So now you have to just see which of them is a viral disease. If you know, you know. So AIDS is a viral disease. It is caused by retrovirus. Ascariasis is caused by a roundworm, so basically Ascaris. Ringworm is caused by fungus, trichophyton, epidermophyton. Amoebiasis is caused by Entamoeba basilica, basically a protozoan. So only one disease is there which is caused by virus, and that is this. So interferons are being released by the cells affected by the virus, and these interferons say the other cells, please be safe, there's an attack happening up over here. Cool? So 8 is the correct answer for this. 8th question. The human host in which the gametocytes of malarial parasites develop are. So, you know what? The malarial parasites have different phases. When they enter, they are in sporozoite stage. There is one stage, okay? Now, the sporozoite stage, they will directly go through a blood to the liver cells. That is the first camp. It's like terrorist, okay? Terrorist, how do they have camp? Similarly, they also have camp. So, liver is their first camp. Okay, where they will get inside, they will divide, they will increase in number, and later the cells will burst open. The cells of the liver, which you call as hepatocytes, they will burst open. Okay, and now the guys which are there, they will be moving down to the next area, that is your RBC, which you also call as erythrocytes. What do you call them? Erythrocytes. So now once it gets into the erythrocytes, what happens? Here also they will divide, and they will form one stage which is known as gametocyte, my dear children. So gametocyte is the stage in which they are going to next produce what? The gametes. But do they produce gametes in the human body? No. They just present. Okay, in the RBC they divide and they form gametocyte, but they do not form the gametes. What happens? These RBCs, when, for example, I am a mosquito, I am a female Anopheles mosquito, I come to suck the blood of the organism. When I suck the blood in, I am going to take in the RBC which is having the gametocyte. Inside my body, inside my gut, that is my mosquito, I am a mosquito, right? Inside my gut, this gametocyte will divide and give rise to what? Gametes. And that's when the fusion and everything happens. So, but where the gametocyte is produced? It is produced in the RBC, my dear children. So, it is erythrocytes where the gametocytes are being produced. In our body, how do they enter? They enter in the form of sporozoites. So there are so many stages, I am not writing all of them. The first camp is liver and the second camp is RBC where they do all the drama. Cool? Chalo. Next. Which of the following water samples in the table given below will have higher concentration of organic matter? Will have higher concentration of organic matter. My dear children, organic matter, is it good over here? No, it is a bad cop over here. So high organic matter means you are talking about high amount of pollution. Please understand that. High amount of pollution. Now for breaking down the organic matter, whom do you need? You need certain aerobic bacteria. Whom do you need? You need certain aerobic bacteria. Cool. This aerobic bacteria are going to act on the organic matter and break it down. But the aerobic bacteria, as the name suggests, they need air which is rich in what? Oxygen, my dear children. So obviously, they will be needing more amount of oxygen also. They will be needing more amount of oxygen. Now, what do you call this requirement as? You call this requirement as BOD, biochemical oxygen demand. Okay, so when there is higher organic matter, that means you need more amount of aerobic bacteria. Now, this more amount of aerobic bacteria will utilize more amount of oxygen. Basically, biochemical oxygen demand will be really high, so that you can break it down properly, the organic matter properly. So, I will say, if you say higher organic concentration of higher concentration of organic matter is there, that means the level of pollution is what? High or low? It will be high. What is the value of BOD? It will also be high. Okay, there is high requirement. If I said the lower concentration of organic matter is there, that means level of pollution is low. Okay, there is less aerobic bacteria to break it down. Obviously, there is less organic matter to break, right? Obviously, in that case, oxygen requirement will also be less. So, value of BOD will also be less. Okay. So, this is the answer. Yeah. Now, the steps of recombinant DNA technology are given below. You have to choose the correct, like in the order you have to say. The first thing that you need to do is, recombinant DNA technology, you need the DNA. So, first of all, you have to isolate the DNA, obviously. There are proteins, there are this, that, so many things are there. So, first thing, you need to isolate the DNA. So, your sixth one becomes the first point. Okay, after isolating the DNA, what you have to do? You have to cut the DNA. You have to cut the DNA, my dear children. Okay, after cutting the DNA, what you have to do? Obviously, one piece won't be enough, so you have to amplify it, you have to increase its number. So, I will say you have to amplify. So, according to this 6, 3 and 2, 6, 3 and 2, 6, 3 and 2, this is the 6, 3 and 2 option. But let's see the next one. So, after amplification, what do? Insertion of the recombinant DNA into the host organism. So, you take this DNA and you are going to insert it into the host organism where it will be dividing itself. After that, fourth one, obtaining the foreign product. Obviously, after putting it in there, it will be giving rise to its protein. So, whatever is there. So, the protein, the foreign product which is there, it will be obtained. After that, the foreign product which is obtained, obviously, it will be mixed up with all the other things which is not needed also. So, you have to do some processing. What do you call this processing as? You call this as downstream processing. Okay, what do you call this as? You call this as downstream processing, and with that we end it. Cool. So this becomes your answer. Easy, no? So step by step you have to go. Sometimes people directly go with cutting of DNA and then isolation. Please don't do that. We have to isolate it first and then only. All this option, no, you have to be really calm, not like Jasmine ma'am. Okay, Jasmine ma'am went so fast because according to Jasmine ma'am, we did these questions. I don't want to waste your time. Okay, so basically what you have to do is, whenever you are dealing with questions like this, go through the points one by one. Okay, just read it peacefully and then look for the terms isolation, cutting, amplification, insertion, foreign product, downstream processing, things like that. Okay. Works? Chalo. Now, 11th question. DNA in a clone of cells followed by detection using autoradiography is called. DNA in a clone of cells. So, DNA in a clone of cells, as in there are so many. So, this DNA which is there, it is actually detected using autoradiography. Autoradiography, that means it is going to show off its color and light and things like that. So, this DNA which is going to find its exact opposite and going to bind with it, my dear children, what do you call this as? You call this as probe. Okay, you call this as probe. So, this probe is going to attach over there, and now wherever this probe is attaching, it will illuminate with the help of autoradiography machine. When you see, it will illuminate and you will know that this is the exact thing that I was looking forward for. For example, I wanted to check, okay, whether this guy is the one who committed the crime. Okay, and from the crime scene I got a piece of segment of DNA. Okay, now I took it as probe, and what did I did? I basically tagged it with something, and then I am putting it in a gel where I have the victims, oh sorry, not the victims, basically I have the suspect's DNA. If I put it over there, this particular probe over there, it will go and match with the it will make a complementary pair with the other one, such that it will glow. And now if it makes a complementary pair, I know that this DNA belongs to the suspect itself, and then I can catch the criminal. This is done in DNA fingerprinting. Okay, so please be sure about it. This is probe. Okay, next, Jiva was growing a bacterial colony in a culture flask under ideal laboratory conditions where the resources sooner or later became limiting, my dear children. Which of the following equations will represent the correct growth in this case? So, there are different type of growth. One is logarithmic growth and the other one is exponential growth. Okay, what happens is that basically in one, you suppose that there is no limiting factor. It will grow like that. But do you think actually it happens like that? No, none of it. Okay, it does not happen like that. And in this case, you get a J-shaped curve. Okay, you get a J-shaped curve. But in actual sense, this is not possible at all. So what happens is that basically there is a S-shaped curve which is being formed, sigmoid curve which is being formed. That means whenever you're talking about the population or whatever thing is there, my dear children, there will be something that will be limiting. There will be a limiting resource. Obviously, they will be all fighting for resources. It will be limiting resource. So, you cannot go beyond a level. Okay, and this particular level which is there, it is known as the carrying capacity. It is known as the carrying capacity which is represented by K, my dear children. So, I'll say the population growth dN by dt would be equal to Rn. R means intrinsic rate of natural increase, the reproductive capacity and everything. R is basically nothing but birth rate minus death rate, okay, which is there happening in the population. And then I can mention K minus N divided by K. This is how much growth can happen within a carrying capacity. Okay, so this becomes an equation. Why do we say this is the equation? Because here you have mentioned that the resources will sooner or later become limiting. When you are saying it is limiting, that means you are talking about this type of growth. Cool. So this is your answer for the question. Now, next question. Which of the following food chains is major conduit for the energy flow in terrestrial and aquatic ecosystem respectively? So, there are two types of food chains, grazing food chain and detritus food chain. Detritus food chain means dead and decaying organisms which die, they are feeded by decomposers and things like that, and whatever things, inorganic nutrients, soluble nutrients which are there, it will be moving out in the soil and the whole cycle repeats. Grazing food chain, as in for example, you will have a producer, this producer will be eaten by a herbivore, okay, and then this herbivore will be eaten by an omnivore or carnivore and things like that. So, you are asked, what do you think is a major flow of the energy happening in terrestrial and aquatic ecosystem? And you might be very shocked to know this, but this is the actual thing. In the case of terrestrial ecosystem, since there are so many organisms which can be dying easily and things like that, okay, and the detritus food chain can be really active. So, I will say that this is the one that is active up here in the case of terrestrial organisms. Since the detritivores which are there, or basically the decomposers also which are there, they need a proper environment for breaking down things, and it is found in terrestrial environments. Okay, in terrestrial ecosystem, my dear children, for decomposers and detritivores, it becomes easy to break down lot of things. Okay, so which is why I'll say this is a major source of energy, conduit of energy. But when I talk about aquatic food chain, okay, in aquatic food chain, the grazing one, sorry, in aquatic ecosystem, the grazing one is more prominent. There also you have phytoplanktons. Phytoplanktons are eaten by zooplankton. Zooplanktons are eaten by small fishes by large fishes, and things like that. And you might feel, why detritus is not much active over there? There it's water is there. There can be the oxygen requirement and things like that will be different. So in that particular case, obviously the decomposition and everything would not be very much possible properly. Okay, it will not be properly happening, which is why grazing food chain is a major conduit of energy flow there. Always remember that. Okay. So this becomes your answer for that. Cool. Next, which of the following is not an example of in situ conservation? In situ as in the place where they are living, they're only conserving them. That is what you mean by in situ conservation. For example, wildlife sanctuaries and things like that. Okay, you're not going to do anything over there. You're not going to live over there. Basically, you're going to let them live, and you are going to move in your own way. Okay, you're not going to pick them out from that location and put it somewhere else. That is what you mean by in situ conservation. They are taken care of in the location that they prefer to be in. Okay, now which one of the following is not an example of in situ conservation? Is it national park and seed bank, national park and zoological parks, seed bank and sacred grove, seed bank and botanical gardens? My dear children, so if we talk about national park, it is basically a conserving in situ only, so I cannot say it is one. It is a one. Okay, when you talk about sacred groves, again it is in situ. Okay, sacred groves, basically I am saving an area because of my religious respects and things like that, but in that way I am also preserving all the organisms up in there. Okay, so in their location only they are being saved. So that is also an example of in situ conservation. What is not? Seed bank and botanical gardens, they are not example of what? In situ conservation. They are ex situ conservation. Seed banks as in preserving the seeds. Botanical gardens as in basically I am preserving the plants. I am taking it from elsewhere and putting it here and doing it. They are not in the natural habitat. Okay, so this becomes your answer. Next question, exploration of molecular, genetic and species level diversity for novel products of economic importance is. So when you're exploring what's all the aspects, okay, of a product, okay, molecular, genetic and species level diversity for novel products, exploration of molecular, genetic and species level diversity for new products of economic importance, what do you call this process as? You call this process as bioprospecting. Okay, what do you call it? You call this as bioprospecting. Now this is something that you have to click in your mind as such. There is no way to remember it with something else. There is no way to understand it with something else. It's just that you have to remember it. So please remember it as such. Okay, there's no other way, that's why. So it comes under biodiversity and conservation topic. Okay, that particular chapter, it's just there. So you have to remember it. Cool? Chalo. Next thing. Fill in the blanks. My dear children, if you feel that I was too fast, I almost utilized 32 minutes for MCQs. Please don't think that I was too fast. I was just trying to explain you the most concise way that I can and also explain you what I am trying to say. So do let me know if you're finding any trouble in understanding. Okay, I have tried my best, but yeah. Now, fill in the blanks question. You have to fill in the blanks using these options that you're having. The interstitial space in seminiferous tubules consists of immunologically what? What do you mean by interstitial space? So, if we say that this is a testicular lobule which is there in the testis. This is what? Testicular lobule. Inside the testicular lobule, you have certain seminiferous tubules, my dear children. So, these are the seminiferous tubules. Now, this space that you see in between, they will be having certain cells. And these cells are immunologically competent cells. What, my dear children? Immunologically competent cells, as in they are responsible for killing any pathogens will be coming through. Okay, please remember that. Apart from that, they also have certain other cells which are known as Leydig cells, which is responsible for the production of hormones, androgens, the male hormones. Okay. Next thing, the version of biogenesis is accepted by majority as the first form of life arose slowly through evolutionary forces from what? From what? So, the version of biogenesis is accepted by majority. Why? Because the first form of life slowly arose through evolutionary forces from non-living molecules. This is a thing which is taken as such from your NCERT evolution chapter. If you see the third page, after below the SL Miller experiment, it is given, this particular statement is given. So it is accepted because it is said that life arose from non-living molecules, not organisms, non-living molecules, and it took some time. Okay, now filariasis pathogens are transmitted to a healthy person through. So filariasis is elephantiasis, where the person will be having big leg and all. Okay, it becomes swollen because swollen lymph nodes are present. Now how it is spread? It is spread by the bite of a mosquito. Okay, it is spread by the bite of a mosquito. What is this mosquito, my dear children? It is Culex mosquito, and whenever I have mosquito transferring things from here and there, I would call this as a vector. I will call this as what? A vector, my dear children. So this becomes the answer for this. So you should know the name of the mosquito. It is what? Culex mosquito, and it is acting as a vector over here. Now Swiss cheese with large hole is produced from. So Swiss cheese with large holes. So large holes are being produced because CO2 is being released like that. So when CO2 is being released, that's when the hole is being created. Now who is responsible for this? This is a bacteria, my dear children. Can you guess the name of the bacteria? Can you guess the name of the bacteria in the comment section right now? Please write 19th. And this is the name of the bacteria. Can you please tell that in the comment section? Let me see who all got it correct. Now this is from the Microbes in human welfare chapter. Okay, now the host cells which have the ability to incorporate foreign DNA within them are called the host cell. Okay, there's a host cell and it is ready to incorporate the foreign DNA. What do you call this cell as, host cell as? Can I say it is competent cells? It can be said as competent cells. What do you mean by that? Basically they are ready to take up things. That is why I will call it competent. It is not recombinant. Recombinant means it has already taken up the DNA, foreign DNA. That means it has a new combination of DNA. That is why it is known as recombinant cell. No, as of now I am saying it has the ability to incorporate. So the cells which has the ability to incorporate, I will call it as a competent cell. Cool. Chalo. Part B contains 2 mark question and you have to answer any 5, but I will be telling about all of them. Yeah. So complete the tabular column given below with respect to the male gametophyte of angiosperms. So cells of the male gametophyte. First of all, tell me what is male gametophyte? Male gametophyte in the case of plants, it is what? Pollen grains, my dear children. Pollen grains will be having an outer exine, okay, which is made up of sporopollenin, and inner intine, which is continuous, and there will be plasma membrane. Now inside this there is a very big cell which is known as vegetative cell. Vegetative cell. And also in the cytoplasm of this vegetative cell there is a small cell which is there, which is known as generative cell. Okay, what is it known as? It is known as generative cell. Now here it is vegetative, here it is generative. Now you are asked about the shape of the nucleus of the cells. Over here there is irregularly shaped nucleus. What is it? Irregularly shaped nucleus. Over here there will be spindle shaped nucleus. Okay, it will be what? Spindle shaped nucleus. Your answer would be looking something like this. Okay, vegetative, generative, irregularly shaped nucleus and spindle shaped nucleus, and with this you are going to get 2 marks. Cool. Next question. Mention the 2 medical grounds on which the pregnancy is subjected to termination. On what 2 reasons we are okay, or the government is okay to terminate the baby, or medical termination of pregnancy is okay. What are the 2 things? First thing, when it becomes dangerous for both the mother as well as the fetus, that's when, or when the fetus has some diseases, gender disorders which cannot be treated really well and it could be problematic for the future, right? Or when it is an unwanted or without the quantities of sex or things have happened. For example, in the case of rape, at this point of time also MTP is being suggested to be done. So MTP is done in the following situation: failure of contraceptive used during coitus or rapes. Okay, that is one thing. When continued pregnancy is harmful or fatal for either the mother or for the fetus. In case of fetus abnormalities, I have given you 3 reasons, and these 3 reasons have to be put down in your answer sheet as well. Please put it down in the form of points. Do not forget the points. Okay, failure of contraceptives, either during coitus, unprotected coitus, or during rapes as well. Okay, now when continuing it would be difficult or will be fatal for both the mother and the child, as well as when the fetus will be having abnormalities. It could be genetic abnormalities or things like that which cannot be treated. That's when it is fine. Okay, now derive the phenotypic and genotypic ratio of a cross between AB blood group parents. Now please my dear children, when I say AB blood group, what is the allele? The allele will be IA, IB. The allele will be IA, IB. Okay, now if I'm putting in Punnett square, this is IA, IB in one parent. Similarly, the other parent will also be having IA and IB. If I cross this with this, if this gamete and this gamete comes together, in a gamete, only this allele or this allele can go. Either one can go, right? So suppose a gamete carrying this and this fused, I get IA, IA. If suppose this and this fused, I get IA, IB. If suppose this and this, again IA, IB. This and this, I get IB, IB. Now you are asked about phenotypic and genotypic ratio, my dear children. Phenotypically, if you see, one is A, this is B, and both of them are AB. So I'll say one is A blood group. Okay, and one is what? B blood group, and two of them are what? AB blood group. This is what? Phenotypic ratio, 1:2:1. What is the genotypic ratio? Genotypically, how do they look, my dear children? So only one is IA. Okay, two of them are IA and IB, as you can see over here, this and this, and one of them is IB and IB. This is the genotypic ratio. Genetically, they look like this. So phenotypic and genotypic ratio is the same, but you have to write it properly. How you can write it properly? You can write it properly like this. So genotypic ratio, if you see, IB is 1, IB 2, and IBB is basically 1. So this is the genotypic ratio. This will be the phenotypic ratio. Write it properly and you get your answer. Okay, write it properly. This is a 2-mark question. Don't lose your 2 marks over there. Okay. Next question. Which sequence of bases transcribed from DNA are found both in hnRNA and mRNA? So basically, whenever a DNA is there, okay, whenever a DNA is there, transcription process happens in DNA. So that's when you get an RNA, but this type of RNA is known as hnRNA, which will be having both the coding sequences as well as the non-coding sequences as well. So what do you call coding sequence? You call them as exons, and the non-coding you call them as introns. Now this is found in DNA, this is as such transcribed in the case of RNA, and this RNA is known as hnRNA. Okay, during transcription you get hnRNA. Now what happens? This hnRNA undergoes a process which is known as splicing. Okay, undergoes a process which is known as splicing, after which what you get is that the introns are being removed as such, and you just get a stretch of what? So many exons coming together, and this is known as mRNA. So now you tell me what is common between hnRNA and mRNA. If you see, the exons are common between hnRNA and the mRNA, my dear children. So which sequence of bases transcribed from DNA are found both in hnRNA and mRNA? It will be exon. Okay, it will be exon. So in a cell, the sequence of bases transcribed from DNA that are found both in hnRNA and mRNA are exons. hnRNA is the primary transcript, the first transcript produced during transcription, and contains both the introns, that is non-coding regions, and exons. During RNA splicing, introns are removed and exons are joined together to form the final mRNA. Only exons remain in both hnRNA and the processed mRNA, while introns are excised and degraded. Write your answer in points. You don't need to write this answer because you have already mentioned that. But please write it in points, otherwise you are going to lose a lot of marks. And tell the teacher what is the thing that she needs to see. Okay, not what you need to show her. Okay, you have to show what she needs to see. So please tell, this is the first, in the first statement only you got the answer, then you are explaining how does it happen. So do that. Okay, and please underline exons and introns. Cool. Now 25th, potato tubers and sweet potato tubers are result of convergent evolution. Justify the statement. What do you mean by convergent evolution? Where there is no common ancestry, there is no recent common ancestry. Okay, and structurally also the things are not same, but the functions is same. The function is same. So if we talk about potato tuber, if we talk about sweet potato, both of them, okay, this is the potato tuber where this is a stem, and the stem is going to give rise to a tuber. This is what? A potato tuber, and this is what? A stem. When you talk about sweet potato, the roots are there and the roots are going to enlarge. This is what? Sweet potato, and this is the root, my dear children. This was what? the stem, my dear children. Okay, now over here, if you see, both of them are doing the common function. That is what? Storage of starch. Storage of starch. But here it is stem and here it is root. The structure is totally different. Do they have a common ancestry? No. So this is an example of convergent evolution. How do you explain this? You explain it like this. Convergent evolution occurs when unrelated species develop similar traits. Okay, due to adaptation to similar environments. So they are both adapted to similar environments. Both of them have to store things, which is why they have the common function. Now potato, which is known as Solanum tuberosum tubers, and sweet potato, that is Ipomea batatas, okay, tubers, both store nutrients but evolve differently. You don't have to name it like this. You can just say potato and sweet potato. Okay, so both store nutrients that is starch, but they have evolved what? Differently, my dear children. Potato tuber is a modified stem with nodes that sprout new shoots. Sweet potato tuber is a modified root for nutrient storage. Okay, it's shoot, it's root. Now, despite their similar function, they originate from different structures, illustrating what? Convergent evolution. So, there is a different structure but similar function. When similar function is there, it is convergent evolution. And what do you call this type of organs? You call these organs as analogous organs. Cool? Works? Chalo. Next, what are biofertilizers? Mention its significance. What do you mean by biofertilizers, my dear children? Biofertilizers basically means when you put fertilizers, they can be of chemical origin, right? And now if you're putting chemicals inside the soil, obviously this is going to cause a huge ruckus in the soil. We don't want that to happen. Okay, so what do we do instead? Basically, if we are providing certain organisms that can capture nitrogen, that can capture phosphorus, that can capture sulfur and provide it to the plant, it will be really good, right? So those organisms which are going to enrich the nutrient quality of the soil, they are known as biofertilizers. And as I said, it enriches the nutrient quality of the soil. That is the significance of this. So how do you frame this? Biofertilizers are microorganisms which enrich the nutrient quality of the soil. Example, you know rhizobium? You might have studied from the 7th or 9th or 10th grade. Rhizobium is something that is going to capture the atmospheric nitrogen. So nitrogen, no, you cannot take it in, plants cannot take it in like that, because nitrogen is like this, N2. There are 3 bonds between 2 nitrogen atoms. So obviously you cannot, the plants cannot come and break it. So for that, what do we have? There are certain organisms, rhizobium in the root nodules, which basically captures this nitrogen and converts into different forms so that the plants can utilize this. Okay, so rhizobium as an example is a symbiotic bacterium that lives in the root nodules of legumes and fixes atmospheric nitrogen into organic compounds that the plant can uptake. Similarly, azospirillum and azotobacter, okay, are free-living bacteria which absorbs free nitrogen from the soil, air and convert into salts of nitrogen like amino acids and enrich the nutrients. Similarly, cyanobacteria, which is a bacteria, you also call it blue-green algae. It's not algae, it's bacteria only. It fixes atmospheric nitrogen and increases the organic matter of the soil through the photosynthetic activity. Now, nostoc, anabena, oscillatoria, all are examples of this, and this is entirely from your NCERT textbook. Is this something from outside? No, it's from your NCERT textbook. So 2 marker question, right? So at least you should be writing 2 to 3 points. In short, you can write, but you have to write. Okay, please remember the names. You have to remember it. Rhizobium, azospirillum, azotobacter. In cyanobacteria, you have nostoc, anabena, and oscillatoria. Because these things are given in NCERT, you can get questions in different format in the form of MCQ also. So why to take a risk? If you're studying a 2 marker question and with this 2 marker question, you're getting 1 more extra mark in the MCQs, why to lose it? Yes? Yes. Now, name any 4 recent extinct organisms as per IUCN Red List. So, again, something directly from NCERT. Dodo, okay, quokka, they are Mauritius, dodo was from Mauritius, quokka, Africa, thylacine, Australia, stellar sea cow, Russia, and 3 subspecies, Bali, Javan, and Caspian of what? Tiger. Okay, dodo, quokka, thylacine, stellar sea cow, 3 subspecies of tiger, that is Bali, Caspian and Javan. Okay, will you be able to remember it? You have to. Okay, we have to remember it by somehow. Cool? Chalo. Part C, there's a 3 mark question. You have to answer any 5, my dear children. Chalo. Draw a label diagram of the fertilized female gametophyte and mention the ploidy of any one of the products of double fertilization. So basically, How do you make the female gametophyte? In the female gametophyte, there will be one egg cell, and surrounding the egg cell there are two synergids, my dear children, having its own filiform apparatus like this. Okay, the synergids. Now here you will be having 3 antipodal cells, and in middle you will be having 2 polar nuclei. So I am just going to name each of them. This is synergid, this is egg cell, this is polar nuclei, and this will be antipodal cells. Now you are asked about one more thing, ploidy of any one of the product of double fertilization. So in double fertilization what happens? One male gamete will come and fuse with the egg cell. Okay, one male gamete will come and fuse with the egg cell. There are 2 male gametes. Male gamete 1 fuses with the egg cell, and what do you get? You get this thing, that is zygote. What is the ploidy of zygote? This male gamete has haploid number of chromosome, this also haploid. Haploid comes together, it will be diploid. So this is zygote, this one. Okay. Now next, one more fertilization happen, but the male gamete will be now fusing with the 2 polar nuclei which is present over here. The 2 polar nuclei, and this will give giving rise to primary endosperm cell. This whole cell, the central cell which is there up over here, okay, which is up over here, this whole cell which has the 2 nuclei, it will now be known as primary endosperm cell, which has the ploidy of 2 and no, my dear children. Male gamete has N, one nuclei will be having N, another nuclei will also be having N. So N, N and then N, you have triploid. What's a ploidy? So these, any of these 2 you can write it. Okay, this process is known as syngamy, this is known as triple fusion because 3 things are coming and fusing together. Together you said 2 times fertilization happened, that is double fertilization. So ploidy of any of the product, either zygote or the primary endosperm cell you can mention. Okay, so the endosperm they have mentioned, it is triploid, and this is the whole thing. So please remember, mention as micro. So this end is known as micropylar end. Wherever the egg is there, no, you call this as micropylar end, and this one will be the chalazal end. It will be the chalazal end. Cool? Chalo. Next. Parturition is induced by complex neuroendocrine mechanism. Comment. So what happens? Basically, parturition means expelling out of the baby. Now this is induced by complex neuroendocrine mechanism, as in both nervous system as well as endocrine system is involved in here. Do you think so? Yes, my dear children. So what happens is that in the uterus, the baby which is formed, the fetus which is there, it will give the signal. The fetus and the placenta is going to give the signal. It is going to release out certain hormones, my dear children, which are known as androgens. These androgens are going to convert into estrogen in here itself. And what does this estrogen do? So basically what happens in the uterus wall, you know, there are 3 layers: perimetrium, myometrium and endometrium. So myometrium is a muscular layer. So when estrogen is released, now what happens? The myometrium will slowly start showing all its receptors up over here. Okay, and these receptors are known as oxytocin receptors. Oxytocin receptors. What is oxytocin? Oxytocin is a hormone, and there are receptors for this particular hormone in the layer, in the myometrium layer. Cool. Now what is exactly happening up over here? Let's see. So estrogen is being released. Now this estrogen is going to basically, okay, estrogen is going to make up this oxytocin receptors up over here in the myometrium. Not make up, basically they help it in expressing itself. And the signal which is being produced like this is known as fetal ejection reflex, which is done by the fetus and the placenta itself. Okay, this is known as fetal ejection reflex. Now what happens? In the mother's pituitary is always releasing oxytocin. So this oxytocin will come and bind to these receptors, and when they come and bind to the receptors, what is happening? There will be muscular contraction happening in the myometrium. So until now the receptors were not active, but now the receptors are active, which is why the oxytocin will come and bind over it and make it contract very much. When it contracts very much, my dear children, because of this, what will happen? Slowly this baby will be going down. It will be coming out. Okay, so please remember. So the act of expelling of the full-term fetus from the mother's uterus at the end of gestation period, as in the whole period when the baby was inside, is known as gestation. Okay, where the whole formation of the baby is happening. Now, the act of expelling is known as parturition. Parturition is induced by complex neuroendocrine mechanism. The signal for parturition originates from the fully developed fetus and the placenta, I told you, right, which induces mild uterine contraction called fetal ejection reflex. So when they are doing so, what is happening? The oxytocin will come and bind, and then this will slowly help in the fetal ejection reflex. This is fetal ejection reflex. There will be small, mild uterine contraction, but the oxytocin will come and bind to it. Now, what will happen when the baby will try to move out? Again, what will happen? Again, there will be contraction. Now, this triggers, okay, the mild uterine contraction, which is known as fetal ejection reflex. Okay, this triggers the release of oxytocin, more oxytocin from the maternal pituitary, and the oxytocin will induce stronger uterine contraction. Okay, so now it is totally hormonal, which is coming, hormonal role that is coming over here. The hormones will again be released, and there will be more and more contraction, and that's how the baby would be moving out finally. Cool. So first, the signal will be coming from the fully developed fetus as well as the placenta. So what happens? There will be mild uterine contraction. Why? Because the receptors are active now, and oxytocin will come and bind on it. That's why there is a uterine contraction. Now, when this mild uterine contraction happens at this point of time, again oxytocin will be released and this oxytocin will come bind again, and then again there will be contraction. So this is a loop that happens clearly. Okay, and this will cause parturition. Fine. Now, the popular and effective contraceptives include IUDs. Mention the types of IUDs with example of each. So, IUDs, intrauterine devices, are something that we put inside the uterus under doctor's administration. Okay, so how do they work? So, intrauterine devices are three different types. One is non-medicated. There is no medicine associated. They don't release anything inside the uterus. They're just there. So, what happens is that lipase loop is an example of this. This is totally foreign for our body. When lipase loop is inserted inside the uterus, the body feels that this is something foreign. So, obviously, it will act on it. Now, when it acts on it, it obviously changes the whole uterine structure over there. Inside the uterus, the whole structure and everything is going to change because it is tackling with something that is foreign. Okay, so obviously anything that will come along with it, for example, at that point of time, coitus has happened. Okay, sexual contact has happened. At this point of time, the sperm that gets in will also be killed, thinking that it is what? Foreign, my dear children, because our body is immunologically really active. This is an example of non-medicated IUD, which is lipase loop is an example of it. The second thing is copper releasing IUD, or you call it copper releasing IUD, which is basically what it does is that copper T, if you have heard of it. So, copper T is basically inside the uterus, it is going to release copper ions, and this copper ions, what do? They mess up with the motility of the sperms. They do not allow the sperms to move, and if sperms are not moving, do you think they can fuse with the ovum over there? No, my dear children. So, the motility of the sperms is being greatly affected, and this happens because Cu ions are being released, and this is the whole principle of the copper releasing IUDs. That's how they work. Now, there is a third one, which is hormonal IUDs. So, example is progesterone and things like that, where basically they are going to release hormones inside the uterus. Now, hormones, obviously it's there in the uterus and things like that. We have progesterone, estrogen and things being released by your ovary. But when their level becomes changed, there is an imbalance over there, it will be not really good for our body. So, initially just think that progesterone was the one that is helping in the maintenance of endometrium, but now since the level is way too much, now they are not going to allow implantation to happen, or basically the endometrium would not be maintained properly. Okay, or implantation, that is embryo will come and slowly implant into endometrium, that would not be happening properly. All these problems will be created. Apart from that, the uterus basically it has an opening over here, not an opening, an entry site over here, which is known as cervix. At this point of time, there will be a mucus plug that will be created such that no sperm can get inside. So, these are the changes that is induced by the hormones. So, non-medicated IUDs, these increase phagocytosis of sperms within the uterus. So, all the sperms which are there, they will be killed because obviously they are also thought to be really foreign. Example is lipase loop. Next, we have copper releasing IUDs. So, along with phagocytosis of sperms, the copper is released, suppresses sperm motility and fertilizing capacity of sperms. Example CUT, CU7, multiload 375, you have to remember all of them names. Okay. Now, hormone releasing IUD, these make the uterus unsuitable for implantation and cervix hostile to sperms. Example progesterone, LNG20 and everything. Cool. This is question number 30, my dear children, and these are the things that you need to remember. Next, write the salient features of the following human ancestors: Rhyopithecus, Ramapithecus, Australopithecus, Homo habilis, Neanderthal man, Homo erectus. All of them basically, it comes in the origin of life in evolution chapter, no? So under that it comes. So Rhyopithecus, my dear children, Rhyopithecus were the first organisms which were there, and they had more ape-like features. When you talk about Ramapithecus, they had more man-like features. Okay, that was the difference between Rhyopithecus and Ramapithecus. So man-like features, Ramapithecus are the ones they further give rise to what human beings slowly. It's not like a one-step process, but slowly happened. Let's read about it. So Rhyopithecus was more ape-like creature. They were hairy, they walked like gorilla and the chimpanzees. Whereas Ramapithecus, they were man-like primates, probably not taller than 4 feet, but walked upright. So not entirely upright, but yes. Okay, they didn't walk like this, but yeah, they were slowly better than the gorilla and chimpanzees one that is present over there. And they were not taller than 4 feet. Now from them came Australopithecus. Now from this guy came the next guy, that is Australopithecus. They hunted with stone weapons, but they essentially ate fruit only, my dear children. Okay, and then came Homo habilis. These creatures were called the first human-like beings. Okay, the brain capacity, if you talk about it, it was 650 to 800 cc. Cc means cranial capacity. They did not eat meat. So they were all like eating fruits and things like that. Okay, and it was all happening in South Africa, my dear children. So Rhyopithecus, more ape-like features. Ramapithecus, more man-like features. Australopithecus, they hunted with stone weapons and they basically ate fruit and things like that. Homo habilis, which are more related to Australopithecus, these creatures were the first human-like features. Okay, they are called as hominid. And the brain capacity was what? 650 to 800 cc. They did not eat meat, my dear children. From Homo habilis onwards, you get the next organism, which is Homo erectus. From here comes Homo erectus. So the Homo erectus, if you see, the brain capacity was greatly increased from 800 cc to 900 cc, and they were the one who started eating meat. Okay, they started eating meat. And from Homo erectus came the next two organisms, which is Neanderthal man as well as Homo sapiens, which is not mentioned over here. So we are the modern Homo sapiens. Okay, but before us we also had Neanderthal man, which arose earlier. So here also the brain capacity increased, and they were the people or the organisms which feel that we have to protect our body, we have to cover it, and we have to bury our dead and all this thing. So Neanderthal man came after Homo erectus, but according to the question, since they have put in like that, we have to go in the order. Okay, so Rhyopithecus, more ape-like feature. Ramapithecus, the term is there, so they are man-like features. Okay, then came Australopithecus, and then Homo habilis, Habibi, the first human being, you can remember like that. The first human-like being features which was there. The brain capacity also was mentioned, 650 to 800 cc. Okay, they did not eat meat, anything like that. Then came Homo erectus, the brain capacity was increased and they totally erect, they stood erect, my dear children, and they had 900 cc brain capacity. They ate meat and did everything. Okay, now Neanderthal man came, they arose from Homo erectus, and their brain capacity greatly increased to 1400 cc, and they used to hide their body, they protect their body and bury the dead as well. Cool. So that is there. Now next, describe any 3 properties of cancerous cells, my dear children. So cancer cell, how it is different from normal cell, you have to think like that. So suppose this is a normal cell, my dear children. So once they start dividing, no, if they come in contact with the other cell, they will stop that division, and this is known as contact inhibition. What do you call this? You call this as contact inhibition, where when they come in contact, they are going to inhibit the further growth. That is what you mean by contact inhibition. Now, that is not seen in cancer cells. Cancer cells, even if they come in contact, they will still keep on dividing. Second thing, if you talk about cancer cells, they show a property which is known as metastasis. What do you mean by that? Cancer cells, for example, here I have cancer cells. These cancer cells can be moving around different parts of my body, and this process is known as metastasis, and they will spread really quick like fire. You get it? And now the third property, they will be creating, okay, cancer cells, if you know, they will be creating their own blood vessels around them so that they can get enough amount of nutrient supply and things like that. Okay, they will be creating their own blood vessels and territory and things like that. So the cells divide repeatedly without uncontrolled cell division. Cancer cells have lost the property of contact inhibition. They invade from one part of the body to the other part through a process that is known as metastasis. Cool? Works? Yes, that is there. Now, Okay, so these 3 points you can write, and also you can write the other point, that is they create their own blood vessels as well for nutrient supply and things. Okay, now complete the below given tabular column with appropriate answer. Let me see if how many questions are there. Okay, 2 more questions from here. So we have to finish this, right? Microbes in human welfare, this is from that chapter. So name of the microbe, name of the product. Lactic acid is being produced. Now lactic acid uses what? It converts milk. It will be converting milk to what, my dear children? Milk to curd. Now name of the microbe which is there, what is the name of the microbe? It is lactobacillus. It is what? Lactobacillus. Now methanogens, what do? Name of the product that they produce. They produce something that is known as methane. Methanogens, they produce a gas that is known as methane, and what is the use? This gas is being used as biogas, my dear children. This is used as biogas. Now again, name of the microbe. Over here, name of the product, treatment of bacterial disease. So this for treatment of bacterial disease. What is the name of the microbe? So I just know one microbe, that is Penicillium notatum. Penicillium notatum, which releases penicillin, and this penicillin is going to act like antibiotic. It is going to treat what? Bacterial diseases. Okay, got it? So here lactobacillus, lactic acid uses what? Uses is to convert, digest the milk proteins. Okay, that is the use. Methanogens, they release methane and it is used as biogas. Penicillin, actually it should be Penicillium notatum over here and penicillin over here. The name of the microbe is Penicillium notatum. It releases penicillin and it is used for treatment of bacterial diseases. Cool? And now next one, represent diagrammatically the pyramid of number in terrestrial ecosystem. How do you represent the pyramid of number in a terrestrial ecosystem, my dear children? So pyramid of number, it looks like this. And terrestrial ecosystem, you know, the number of plants and everything is way too much than other organisms. So obviously the producers, T1 will be more, they are the producers. Now T2 which is there, it will be less. Okay, for example, producers are being eaten up by rabbit. Okay, now rabbit is being eaten up by tiger. The level of tiger will be way too less. So in terrestrial ecosystem, what can I say? The amount of organism which goes up will be really less. So it will look like this. This is from your NCERT itself. So 5,84,000 to 58,42,000 organisms are there in primary producers. Then primary consumers are there, which occupies T2 level. They will be less. Again, secondary consumers which they will be comparatively lesser. Tertiary consumer, again comparatively lesser, 3 number and things like that. This is the case of terrestrial ecosystem. Okay, the pyramid of number, it looks like this. Okay, and now we have come to part D, that is 5-marker question, and you have to answer only 4. But before entering into it, I want to say something. The answer for the question, that is Swiss cheese, okay, it produces CO2, right? It produces CO2. In Swiss cheese, the CO2 is being produced, which is why there are large holes. What is the name of the bacteria which is responsible for producing it? Its name is Propionibacterium shermani. That is the name, Shermani. That is the name of the bacterium responsible for it. Okay. So if you are missing out on that, please don't miss out on that. Yes. Now going ahead with the next question. Flowering plants have developed many devices to discourage self-pollination and encourage cross-pollination. This is a 5-marker question, and you can take 1 minute break and come fast really fast. I will also take a 1 minute break and come fast really fast, but in a little amount of water. Okay. Okay, so here I am with my bottle of water. I hope you drank yours as well. Okay, so we were talking about the flowering plants, okay, and how do they discourage self-pollination? First thing, you have to know why do they discourage self-pollination. Now you only think self-pollination is basically what? The male gamete and the female gamete are belonging from the same plant. If they come together, obviously there is no chance of any big variations and things to happen because both of the cells are from the same plant. So same genetic material is coming together. So whatever defects were there in this plant, the defect will be there. Okay, there is no newness over here. Okay, there is only a bit amount of newness that is not much needed. Okay, we need lots of newness. So, which is why self-pollination is actually very much discouraged. Okay, and it causes something that is known as inbreeding depression in plants. Okay, and in animals also. So, basically what happens is that offsprings which would be produced that would not be fertile and stuff like that. So, we don't want that. And we want to encourage cross-pollination where the male gametes from one plant is going to come over here to the female gamete of another plant. So, that means two genetic material has to come together. So, what have the flowering plants done for doing so, for preventing self-pollination as well as encouraging cross-pollination? So, the first thing that they have done is producing unisexual flowers. Now, you only think if unisexual flowers are there, my dear children, okay, in a flower there is only one sex, that is probably the stamen. So, obviously, the male gametes from here have to travel down to the other flower which would be having the carpel. Now, again, if they are on the same plant, is the same genetic material, but suppose they are on a different plant, then suppose they are on the different plant, then obviously, don't you think it is doing cross-pollination and is going to give rise to more genetic varieties? Yes. So, that is one thing that has been done. Production of unisexual flowers and also unisexual plants. Okay. And the next thing, it's not necessary. Sometimes what happens, suppose this is a flower and in this flower you have the female reproductive part and the male reproductive part. Suppose this guy is maturing way early than this, then the pollen grains which are released over here are not going to land on the stigma and basically produce a pollen tube over there, because the stigma is not yet receptive to take this pollen grain and make a pollen tube over there. You get it? So, stigma receptivity as well as pollen maturation of pollen sometimes are not synchronized. Okay, it's not synchronized in a way that both of them basically cannot produce a fertile offspring, which is a proper plant. You get it? So, that is one more thing that is there in the case of plants. Now, apart from that, plant itself has something, there's a genetic mechanism to stop this particular thing, that is self-incompatibility. Incompatibility. What do you mean by self-incompatibility? That means the pollen grains from here, suppose it landed on the stigma, but the stigma says that I am not ready for giving pollen germination. I would not allow pollen germination. This is totally you're totally incompatible. I cannot do so. So, there are some genetic differences which are being made because of some changes in the genetic makeup of the plant, which is why they do something that is known as self-incompatibility, my dear children. So, these are the three very important devices which is there for discouraging self-pollination. When you read it, it becomes all the more clear. So, unisexual flowers, as I said, if both the male and the female flowers are present in the same plant, such as castor and maize, it prevents autogamy, but not geitonogamy. Geitonogamy as in still self-pollination will be there. Autogamy as in, for example, if this is the female and this is the male, from here to here if it goes, this is known as autogamy. Now, suppose it's like this. There's a female part over here and there is male part over here. So, from here to here again it's on the same plant, but between two different flowers. What do you call this? This is known as geitonogamy. So, it is said in unisexual flowers, autogamy will be prevented obviously, but no, geitonogamy would not be prevented. What do we have? One more option we have, male and the female flowers are on separate plants, on two different plants. You call this as dioecious condition. You all call this as dioecious condition, and this will prevent both autogamy as well as geitonogamy. Example, papaya, my dear children. So, have unisexual plants, that is better option. So, one option is having unisexual plants. Okay, that means they are both dioecious. They have only one sex in one particular plant. Okay. Now, dichogamy is something else. What do you mean by dichogamy? Basically, in some species, the pollen release and stigma receptivity are not synchronized. It is not synchronized. Stigma is not receptive, still the pollen is being released. That means it's not going to work. So, to prevent self-pollination, this is being done. It is known as protoandry when the flower in which anther mature earlier to stigma to prevent self-pollination. Here, the anther is maturing first. Protogyny, where the stigma is going to mature first rather than the anther. Okay, so dichogamy is there. Under that you have protoandry as well as protogyny. Now you have something that is known as self-sterility or self-incompatibility. What do you mean by that? The pollens basically do not germinate on the stigma of the flower due to mutual inhibition. Okay, and this is totally genetic. This is a genetic mechanism that prevents pollen grain germination on the stigma. So basically, in short, you have three mechanisms. One is this, second is this, and the third one is this. Okay, here unisexual plant, that is dioecious plant, example papaya and things like that. Then you have dichogamy, where the pollen release as well as stigma's receptivity is not synchronized. It is protoandry when the anther will mature first, and protogyny where the stigma is going to mature first. Okay, and then there is self-sterility or self-incompatibility, where the pollen grain do not germinate on the stigma of the same flower due to what? Mutual inhibition, my dear children. Got it? Chalo. Next thing. Draw a labeled diagrammatic sectional view of the human female reproductive system. So this is the diagrammatic sectional view that you have to draw. I don't think I have to draw it and show you. If you think so, I can draw it. No problem. Okay. So basically what do? You make something like this, and then you do like this. Wow. Okay. This is how I remember. Okay. Now, this has to have finger-like projections like this, and then something like this. Similarly here also, certain finger-like projections and something like this. Over here, you come. Okay, I made something very bizarre. Just give me a second. Okay. So if I make something like this, will it be fine? Comparatively fine, I think. And now, cool. Or probably I should just erase and draw it once more. I don't know, sometimes when I draw like this, it becomes bad. Sometimes it becomes really, really good. Probably I should be making a small diagram for that. Okay. And here also. Works. Okay. See, the thing is, I am getting light reflection and you are not, which is why I am not able to draw properly, but I don't think you will be having that problem. So this is the ovary. Here also you would be having the ovary, my dear children. Okay, do not draw like this. This is just for you to understand certain things. So this is the ovary. This area that you see is known as fimbriae. And this belongs to what? Infundibulum. This is ampulla. And this is what? Isthmus. Cool? Now this will be the vagina. And over here, my dear children, you will be having cervix. Cool. Now it looks better. This is the uterine fundus, and this is your whole uterus. So this is the female reproductive system, okay, in human beings. Draw it properly, not like this. I know I made it beside because the other side I'm not able to see because there are so many tube lights being arranged for the lightings and stuff like that. Okay, so I'm not able to entirely see the diagram, which is why I do like that. But I hope you have just mentioning all the important things that is mentioned up over here. Okay. So just for you to give, what say, a company, because a lot of you might think that you don't know to draw better, and that's why you're not gonna draw or you're gonna mess up. It's for you, my dear children. Do not lose. I am drawing in front of thousands of people like this. Being a teacher, so obviously you should be having the courage to draw it by yourself, being a student. Okay, so I hope it helps. So all this important part, my dear children, each of it you have to remember and you have to draw. Going ahead with the next question that we have. Few autosome-linked recessive gene blood diseases occur in human population. Among them, some are related to qualitative and quantitative problem of synthesizing blood proteins. Example, explain. My dear children, so few autosomed linked recessive disorder, as in what? Basically, it is not sex-linked disorders. Okay, it is just on the other chromosomes. There is some mutation in the other chromosomes, not your sex chromosome, some other chromosomes, and it is a recessive condition. That means when both the alleles are recessive, that's when this particular disease is being shown. Okay, this particular disease is being shown in that population. Okay, now now mostly it is related to what? Blood, related to synthesizing blood proteins and stuff like that, and you have qualitative and quantitative diseases like that. So I'll talk about two diseases which is related to blood. One is sickle cell anemia and the other one is thalassemia. So what happens in sickle cell anemia? You know, in hemoglobin there is alpha chain and beta chain. So over here the beta chain is getting affected. Why? Because there is a gene, that is HBB gene. Okay, this gene has some mutation in it. Okay, has some mutation in it. That is, the codons which are there coding for amino acids in this particular gene. Okay, suppose it was GAG. Now there is a replacement at A position, and now it is what? GUG. This is the 6th codon. Okay, we have different codons in mRNA, my dear children. This will be the 6th codon, and the 6th codon A is replaced by U, which is why this amino acid, which was glutamic acid, is now what? Valine. And I hope you know that all these amino acids, they come together to form what? Protein. To form what? Protein. And what is this protein? This is the beta globin chain protein that we are going to make. And this beta globin chain protein is coded by what? HBB gene. Suppose if this gene has some mutation, which is why, okay, the mutation is what? The A is replaced by U. Okay, and now it is coding for valine. So what happens? The beta chain which is being produced, okay, and the structure of it is quite different, which is why the RBC carrying it, the RBC carrying this particular defected hemoglobin, my dear children, defected hemoglobin will be spindle shaped. It will be what? Spindle shaped. Now you only think, is it a qualitative error or quantitative error? It is obviously qualitative error. The quality of this particular RBC is being lost, which is why, since it was discoid, usually what is the shape of the RBC? It is discoid in shape, which looks something like this. So you have to make a round like this and make another round. So there's a small bend over here. This is known as discoid shape. But as of now, it is spindle shaped. And now, do you think it can carry enough amount of oxygen and all? No, Baba. It cannot carry enough amount of oxygen. Okay, so it is not HBB gene. Basically, the gene's name is HB. Okay, so normally it will be HBA. It will be HBA. If this is a condition, then the person is totally normal. Now, when sickle cell anemia is there, if someone is a carrier for sickle cell anemia, they will have one HBA and the other one will be HBS. This is what? Carrier. But now this is an autosomal linked recessive gene disease. So both of the genes should be recessive. That's when the disease is shown. So now it should be HBS, HBS. That is when the person will be affected with sickle cell anemia. Here the person is what? Basically just, okay, it is basically just the carrier. Here the person is affected, and in this particular case, my dear children, okay, so rather than writing this also, this would be also wrong, rather than writing this also, so condition when it is like this, that's when your hemoglobin beta chain is affected, which is why you write HBB. Okay, and with that you understood sickle cell anemia. Now what about thalassemia, my dear children? Now I told you there is alpha chain and beta chain. In thalassemia what happens is that alpha chain, there are alpha chain which is there, it is coded by two genes. Okay, it is coded by two genes. One is HBA1 and the other one is HBA2. Okay, it is found here also, HBA1 and HBA2. Suppose any out of these four genes, any one is affected. Basically, it is going to affect the quantity of the alpha chain which is going to be produced. Now, for beta, there is only one gene, that is HBBB. Suppose any of these two genes are affected, it is going to affect the quality of the, sorry, not the quality, quantity of the beta chain which is being produced. So this becomes a quantitative disorder. Suppose two genes are getting affected, two alleles are over here are getting affected. That means what? My alpha chain totally is getting affected. Only one, the other two alleles are left. So this is drastically affected. The quantity of it is greatly reduced. The alpha chain is greatly reduced. Similarly, if both of them are affected, that means beta chain is not being produced. Again, quantity is being reduced. So you have major, thalassemia, thalassemia minor, depending on how many genes are being affected. Okay, and this is a quantitative disorder. And you can understand it better by reading this. So listen to me carefully. Some blood diseases in humans are caused by mutations in autosomal recessive genes, leading to issues with synthesis of blood proteins. These problems can be categorized as qualitative, affecting the structure or function of the protein. That is, you're talking about beta globin chain, that was getting affected. So the structure and the function is getting affected. And quantitative, affecting the amount of protein which is being produced. So in sickle cell anemia, which is a qualitative problem, mutation in HBB gene on chromosome number 11 causes the production of abnormal hemoglobin, that is HbS. It produces what? Abnormal hemoglobin, that is HbS. Now, this defective hemoglobin leads to sickle-shaped red blood cells, impairing oxygen transport and causing blockages in the blood vessel. Here, the defect is caused because of error in the quality of the protein which is being produced. So over here, I want to just make a quick change up over here. So I'll just write HbA, HbS genes are being involved rather than writing HBB gene, okay, or I can say mutations in the gene coding for beta globin chain. That will be more correct. Okay. Now, this where is it? Okay, now thalassemia caused by mutations in gene responsible for hemoglobin synthesis, HbA1 and HbA2 for alpha chains and HbB for beta chains. I told you there is only one gene for beta, and that is this. There can be mutation in one allele of a gene or both the alleles of a gene, which leads to reduced or no production of specific hemoglobin chains, causing anemia, fatigue. Here the defect arises due to what? The quantity of the gene that is being produced, my dear children. So please be sure about this. This is a qualitative disorder, this is a quantitative disorder. Okay. Rather than writing the whole thing, you can just say mutations in the gene coding for beta globin gene causes the production of abnormal hemoglobin. That's all. You don't need to write HbS and things like that, or this also. Just need to write this. That's all. Close it up. Okay. Cool. And the alpha chain, okay, this particular genes are present on chromosome number 16, whereas beta, this is present on chromosome number 11. So here chromosome number 11 is getting affected, here also this is 16. HbA1 to HbA1 is present on 16, whereas this is present on 11. Cool. Now going ahead, DNA replication is fast, accurate, energetically expensive, substrate and enzyme dependent, initiated from specific site, and cannot uncoil on its entire length. Explain the process of DNA replication considering all these factors. So you just have to mention about DNA replication, nothing else. Don't think about how can I include all these points. Just tell whatever you know about DNA replication. So you have to tell about the enzymes which are involved and the steps. First is initiation, then is elongation, and then is what? Termination, my dear children. In initiation, you can initiate replication only from one side, that is what? Origin of replication, my dear children. So when I say initiated from specific site, isn't that condition being fulfilled over here? Begins at origin of replication. Now helicase unwinds the DNA. There is one enzyme that will be uncoiling the DNA, my dear children, at specific area. At specific area, it will be uncoiling the DNA such that the DNA would be looking something like this, like a fork, which is present over here. Can I just uncoil the whole DNA? No. Why? Because they have been bonded by hydrogen bonds. It will be too energetically expensive if I do so. So helicase basically unwinds the DNA at only specific site, hence forming the DNA replication fork, which because uncoiling the whole DNA is energetically expensive. Now mentioned over here, it is energetically expensive, and also it is substrate and enzyme dependent. So isn't this enzyme dependent? It is dependent on helicase, right? Now still you will see more enzymes. Now single strand binding proteins stabilize unwound DNA. So what happens? Basically there will be proteins coming and binding over them such that they cannot come and bind again each other. You get it? And this is why it is said substrate dependent also. So single stranded binding proteins, they are substrates, okay, which are basically going to stabilize the unwound DNA. Now primase synthesizes primers to provide a starting point for DNA polymerase. Now what will happen? There will be primase which will be giving out certain nucleotides like this, and these nucleotides are going to help with what? DNA replication to begin with. So DNA polymerase will bind on these primers and then only they will start elongating this. So first you need primase to create the primers, then DNA polymerase will come and elongate it, my dear children. So primase is another enzyme that is needed. Now elongation. DNA polymerase synthesizes the new strand in 5' to 3' direction. Okay, it is accurate, it is fast. Okay, it is going to synthesize in 5' to 3' direction. Leading strand is synthesized continuously. That means the strand which is like this, which is in 3' to 5' direction, in this strand the DNA synthesis happens really fast because DNA polymerase works in 5' to 3' direction. But in the other strand, which is in the opposite side, okay, 5' to 3', here the things would be quite different, my dear children. Here they will be, DNA polymerase cannot create copies like that. Oh my god, just a second. Okay, it won't be able to create copies like that. Why? Because it is in the opposite direction. So it will need some help in that. Okay, so it will need Okazaki fragments. So there will be primers attached like this, and from these primers there will be elongation happening and things like that. So there is Okazaki fragment formation over there. Later what happens? All the strands which is there, it is being joined together by another enzyme which is known as DNA ligase. Cool. Now, proofreading by DNA polymerase ensures accuracy. There is another DNA polymerase which will proofread everything. That is, whether I bound A with T only or if I have bounded A with G or something else. So that has to be checked. So who does that? Proofreading is done by DNA polymerase, which ensures accuracy. Now I told you this is accurate because of this. Okay. Now termination occurs when replication forks meet or reach termination sequences. Okay. When suppose from replication is happening from that end, this end, now replication fork are coming and meeting, and this point termination will happen. Or there are termination sequences also. If they meet that, then also termination will happen. Enzymes remove RNA primers and gaps are filled by DNA polymerase. So these RNA polymerase primers which are being put, those are being removed by the enzymes and the gaps are being filled. DNA ligase seals the final nicks in the backbone. Nicks as in the gaps which is there, it has been filled by the DNA ligase. Cool. So this is how you have to explain all the points, my dear children, and you can also mention what is the speed of DNA polymerase as well. 2000 base pairs, okay, per second in the case of E. coli, this prokaryotes and stuff like that. Explain that and put it up there. So that means you can explain how it is fast. Okay. Yes. Now, some drug bottles had their name labels missing in a drugstore of a hospital. The staff needs to identify the drugs with their actions still written on them, analyze their actions listed below and identify the name of each drug and also the source of each one of them. So, used by doctors as sedative and painkiller. What is that drug which is used as by doctors as sedative, my dear children, and painkiller? Morphine. Okay, it is an opioid. It is an opioid and it is what? Morphine. Sedative, you have to remember like that. Okay. Helps patient to cope with insomnia and depression. Now this is done by barbiturates, amphetamines, amphetamines and stuff like that. Okay. Increases blood pressure and heart rate of consumer. If you eat this, your blood pressure and everything will be increased. What is this? This is nicotine, my dear children, which is there in tobacco. Now, act as depressant and slows down body function. It will depressant, it will slow down all your body functions, my dear children. Again, an opioid just like morphine. In this category comes another drug which is heroin. It is what? Heroin. It is a depressant. Now, affects cardiovascular system of the body. Cannabinoids, okay, marijuana, ganja, all of them, they are going to affect the cardiovascular system. So there is only one drug given in your book that affects the cardiovascular system, that is your heart, your blood vessels and things like that. Everyone else is affecting the CNS, central nervous system and things like that. Okay, so that one thing is cannabinoids. Okay, cannabinoids taken from cannabis sativa, this ganja, marijuana and things like that. Okay, so how do you answer this? You can answer like this. I have put on all the answers up here. You can just tally your answers. Please remember, sedative, painkiller, morphine. Okay, depressant, always heroin. And all the other thing. Insomnia and depression, again, barbiturates, amphetamines. Insomnia as in you are not able to sleep and things like that. Okay, that is insomnia. To treat that, you need them. Now, blood pressure, nicotine. Okay, cardiovascular system, cannabinoids. Now, transgenic animals provide innumerable benefits to human beings. Justify the statement with common reasons. So how do you say? Transgenic animals are basically the ones which have some recombinant DNA in them, okay, like us, like our DNA has been put in them and some things are being made out of it. For example, I want a protein that is alpha-1 antitrypsin. Now this protein is really important for me to treat emphysema, which is a disease, a respiratory disease, okay. Now if alpha-1 antitrypsin is being produced by some other organism and I am able to use it, then why not do it? So basically for production of biological products, I can use transgenic animals, my dear children. I can put the gene in them and make them produce it, okay. In the case of, there was a cow that is Rosie. It's a transgenic cow. It was producing a milk that is rich in human protein, that is alpha-lactalbumin. Now over here it is able to produce that, okay, it is able to produce that because I am putting the gene over there and I am getting total benefit of it. Then why not? Now apart from that, apart from using them for biological products, I can also use them for testing the safety of vaccines. So vaccines I can put on them and then basically I can see if they are able to survive it, I can put it on me. Now this is a very bad thing to do, but this is how it works. Okay, apart from that, there is chemical testing. That means, for example, I do not know how much chemical will be bearable by my body, even though I want to treat my body with this chemical, okay, but I don't know how much will be okay for me. So I can check the toxicity level by putting it some other organism and see their effect on it. You get it? Okay, so that is one thing. So basically, can I just put it into points? First thing, if I want to study the normal physiology and the development of an organism, okay, what gene does what? So if I put that gene and see that functional change and things like that, I would be able to understand that. Study of any disease, for example, cancer and all, if I want to study, what do? Basically, I put the same medicine in those organisms and see what are the results of it, okay. Biological products, I also told you about all the things, okay. Vaccine safety and the chemical safety testing. Just go through it and this is how you have to frame your answers. So 5 marker question is there, 5 points have to be mentioned, but you don't have to write this much. This is just for you to understand it, okay. Just don't dig deep and just write so much. No one is going to read your answers like that. It's so boring, okay. Now getting ahead with the next question, and we have Aishwarya ma'am, so we will be tackling of it in some other day, okay, but I will be doing it and I will be putting it forward and you have to tell me the answer for the Swiss cheese question, okay. So now it's time for 41st question. Explain the fascinating forms of interactions in brood parasitism and sexual deceit. It is actually very much fascinating. I'll tell you how. So you know about cuckoo and crow, right? So what happens is that this papa crow, it lays its egg in its own nest. Now cuckoo have evolved in a way that it has started laying eggs which is kind of very similar to the crow's egg. It slowly goes in and sneaks in and puts his egg over there. Now what is happening? Why do I call this as parasitism? Because basically this particular parasite, which is the cuckoo, is giving the egg over there, and this egg, its own egg, is getting nourishment from some other organism, and that other organism is basically should be using all of its energy into taking care of their own kind, but isn't it actually providing some energy for the nurturing of that particular egg as well? So don't you think that it will be known as parasitism? Yes, this is parasitism. And always remember, it's not the prey who have evolved, it's the parasite who has evolved, basically, my dear children, in a way that they lay the same type of egg, just like the poor little crow. Okay, so that is what you mean by brood parasitism. Now what is sexual deceit? Now that is something else. So you know, Mediterranean orchid is an orchid office. What happens is that it looks exactly like the female version of a bee. So the male version of the bee comes and tries to pseudo-copulate with this particular female, but actually, can it copulate? No, Baba, it's not a bee, it's just a plant. And why is it doing so? Why is this plant being so mean? Because it wants to give away all its pollen grains to the bee. Now the bee, when it comes and try to copulate, obviously it will get off all the pollen grains, and now when the bee goes to another plant, it will pass down these pollen grains. So that is a way in which they live. Again, that is really, really bad, but this is how it works. Now let's see, how do you frame the answer? So brood parasitism in birds is a fascinating example of parasitism in which the parasitic bird lays its egg in the nest of its host. Okay, you do not call it prey, so sorry, you call them host, and lets the host incubate them, as in take care of them, provide them warmth and everything. During the course of evolution, the eggs of the parasitic bird, the cuckoo, have evolved to resemble the host eggs in size and color to reduce the chances of host bird bird detecting the foreign eggs and ejecting them from the nest. Obviously, if it was different, the host egg will be like, huh, who is this? I'll just kick it off. So that's why they have evolved. Now, talking about sexual deceit, the Mediterranean orchid orchis employs sexual deceit to get pollination done by a species of bee. One petal of its flower bears an uncanny resemblance to the female of the bee in size, color and markings. Now, the male bee is attracted to what it perceives as a female and pseudocopulates with the flower. And during that process, it is dusted with the pollen from the flower. When the same bee pseudocopulates with another flower, it transfers pollen to it and thus pollinates the flower as well. Cool? So that is the whole story. Going ahead. Now we have again 5-mark questions, but we have to answer only one, but obviously we would be answering all of them, but let's see what are the questions. Now those are some tricky questions which are being put up over here, but it is interesting as well. So results of Mendelian dihybrid cross are represented in the form of Punnett square. Answer the following questions with respect to the results of the dihybrid cross. You have to find the number of parental types progeny. What are the parental types progeny, my dear children? Okay, parents were what? As you can see over here, they are yellow and round, and green and wrinkled. Okay, capital Y for yellow, capital R for round, small y for wrinkled, small y for green and small r for wrinkled. So this is round and yellow and round, and this is what? Yellow and wrinkled. Those are the parental types. So we have to find who are the parental types, my dear children. So you know 9 and 1. Okay, I don't have to calculate everything. So 9:3:3:1 ratio, you know the phenotypic ratio, right? Among them, 9 of them are yellow and round, and 1 of them will be what? Green and wrinkled. Okay, they will be just like the parents. So dominant type of progeny are 9, which is yellow and round, and the 1 is representing the recessive parent, which is green and wrinkled. So how many of them are of parental types? 9 plus 1, my dear children, 10 of them will be parental type. Okay, 10 of them will be parental type. Now, number of recombinant progeny, something that is not found in the parental generation. Now, this is new combination. So, yellow was with round, green was with wrinkled. Suppose, now you have this thing where yellow is with not round but wrinkled. 3 of them are yellow wrinkled, and basically 3 of them are green round. So green and round was never a combination, but now it is. So green and round. 3 plus 3, my dear children, 6 of them are recombinant progeny. Cool? Now next, number of homozygous recessive progeny. Homozygous recessive. Homozygous as in both the alleles should be recessive, my dear children. So homozygous recessive, totally recessive, I just have this one. What is this? If you cannot see, I can zoom it and show it to you. Small y, small r, small r. None of the other places you will be seeing both of them to be recessive. It's totally recessive. So the question says number of homozygous recessive progeny. Number of homozygous recessive progeny as in totally it should be recessive, and there is only one such progeny, my dear children. Okay? Okay. Now, number of homozygous dominant progeny. Now both of them again should be dominant. You only see. You only look over here and tell me both of them, how can they both be homozygous dominant? Where can you see them? Both the alleles to be dominant. Only one again over here, capital Y, capital Y, capital R, capital R. So again, the answer will be what? 1. Now, number of homozygous progeny for both the traits. Number of homozygous progeny for both the traits. For both the traits, how many of do you think they are homozygous? For both the traits, only 2 of them are like this, no? Why capital, this one, both the traits it is homozygous, and then this one, none of the others are. So what can I say? It's 2, just 2 in number, 1 plus 1, basically 2, right? Let me see if all the answers are correct or not. 10, 6, 1, 1, 2. So 10, 6, 1, 1, 4. Okay, yeah, I knew I'm gonna mess up, which is why I put the answers. So don't think that I copied the answers, I only found the answers also. But sometimes in pressure, we forget things. So my dear children, homozygous for both the traits, right? So this capital Y, capital Y, capital R, capital R, homozygous. No, over here. Capital Y, sorry, small y, capital R. Again, homozygous, they are both same for both the traits. Okay, so 2. Now, This one, oh you, yeah, this one, capital Y, small r, smaller. So this is 3. And now this one, small y, small r. So if you cross like this, you get all the homozygous one. So it is not homozygous progeny for both the traits. It is not 2, it's 4. Okay. Okay. Now number of heterozygous progeny for both the traits. Heterozygous for both the traits, my dear children. Again I wrote 2. Oh my god, it's 4. Okay. Number of heterozygous progeny for both the traits, it is, should be heterozygous for both the traits, my dear children. So it's homozygous. This is homozygous for one trait and heterozygous for another. I have to find heterozygous for both the traits. It should look like something like this. So this is heterozygous. In here, this is heterozygous. In here, this is heterozygous. In here, this is heterozygous. So if you see, we are heterozygous for both the traits. How many of them? Basically 4 of them. Okay, again 4. Cool. Now number of pure line progeny. Pure line progeny means you're talking about the same thing over here. Okay, you're talking about them, both of them. This is the pure line progeny that is there, just like the parents. So 2 of them are there. So I'll write 2. Okay, 2 of them are there. So I'll write 2. Now number of homozygous progeny for a single trait. For a single trait, there should be what? Homozygous, my dear children. So we have to find out. Chalo. This is going to be really critical. So over here, this is homozygous for both the traits. This is homozygous for one trait. This is not. This is heterozygous, right? So this is 1. Okay. Again, homozygous for one trait. 2. This is 2 now. Homozygous for one trait. 3. Okay. Homozygous for one trait, because this is heterozygous and this is homozygous. 4. Homozygous, sorry, homozygous for one trait. Okay. Again, homozygous for one trait. 6. Now I don't have hand. Okay. Homozygous for one trait. Homozygous for one trait. How many? 8. I have 8 of them which are homozygous for one trait. Why? Whenever you see the same alphabets together, okay, same alphabets together, they are homozygous. But we have to make sure it's only for one trait. So this is for one trait, my dear children. The other one is heterozygous. Here also, for other trait, it is heterozygous. But this is for homozygous. So how many of them do you have? You have 8 of them like this. Ayo, yes. You have how many, my dear children? You have 8 of them which are homozygous for a single trait. Cool. Now, number of heterozygous progeny for a single trait. Same thing if I find the opposite version of it, my dear children. Heterozygous progeny for a single trait. So here the same thing. This were homozygous for this. That means the other one were heterozygous. So same count will be there. There won't be any change in the count, right? So let's see. Anyways, so this is heterozygous 1. This is heterozygous 2. Okay. This is heterozygous 3. And this is heterozygous 4. This is heterozygous 5. Ayo. Yes. This is heterozygous 6. This is heterozygous 7. And this is heterozygous 8. Again, 8 only. For you guys only, I did it. But I'll tell you the truth. For myself also to check once more, I did like that. Okay. Now, number of recessive progeny for single trait. You know, human beings are always prone to make mistakes. So, even I can make mistakes. It's quite common, right? But I don't want you to get the cost of my mistake. That's why I just reject. Now, number of recessive progeny for single trait. It should be recessive. It should be recessive for a single trait, my dear children. Tell me. Recessive for a single trait. Number of recessive progeny for a single trait. This I have to check once. 6. I have not mentioned. Okay. Anyways. No. What is happening? Okay. Sorry. So, we have to count the recessive ones. So, 1, 2. Okay, wait. 1. Okay. 1, 2, 3. Recessive progeny for a single trait, no? Wait, I messed up, wait. 1, 2, okay, so yes, we were talking about number of recessive progeny for single trait. Now, I had to check a bit, but yes. Listen, over here, this one, if I try to zoom in, come on, okay. Small y is there. This is recessive. For one trait, there is no other things recessive, right? Here also small y is there, recessive. 1, 2, and then here if you see, small y is there, recessive. But it's not recessive over here, so it will be just recessive for a single trait. So 3 of them are recessive for a single trait, that is small y. Now talking about smaller, over here smaller is there. This is not a recessive, okay, this is dominant over here. Again recessive over here, again recessive over here. Why can't I count this? Because both of them are recessive. I don't want that. Recessive for a single trait is being asked. So 1, 2, 3 for y, small y. 1, 2, 3 for smaller. So what is the total number? 3 plus 3, my dear children. It will be 6. Okay, it will be 6. It's kind of tricky, but yeah, it's not very difficult. You just have to stay calm and try to find the answer, then only you can. Okay, so over here is a list of the answers. If you want to just cross check or things like that, you can find it over here. Yeah. Now, the next question, given below are sequences of nucleotides in a particular mRNA and amino acids coded by it. Write the properties of genetic code that can be correlated from the above given data. Okay, so first thing, I see AUG over here and AUG is for methionine. So, I know one thing about AUG, that AUG is going to act like a start codon. It is going to act like a start codon, and it is also the one that codes for an amino acid, that is methionine, my dear children. Apart from that, if I talk about UUC, both of them, they are different codons. They are different codons, but still they are coding for phenylalanine. Now, this property is known as degenerate property of codons, where different type of codons are coding for the same amino acid. So, over here you saw UUU, okay, UUU and UUC, both of them they are coding for what? Phenylalanine. Now, if you go ahead, okay, what is being missed over here? Okay, actually GUG is for valine, that is being put over here. So, UAA, it is not coding for anything. So, UAA is not going to code for anything. That means there are stop codons as well. These are what, my dear children? Stop. They are stop codons. They do not code for anything. Just terminate the process of translation. Okay. Apart from that, what do you have? For each and every codon, you have an amino acid. That is one feature that is there. Apart from that, do you think that there is any full stop, comma and things like that? No, Baba. It's all continuous. The whole sequence that is being put over here, it is all continuous and it is being continuously. So that is one more feature. Next thing, it's common for all. In all the organisms, mostly UUU stands for phenylalanine only. So this is also one feature which is up there. Now, how do you frame your answer? You have to put your answer like this. Basically, the codon is triplet. Yeah, if you saw all of them, my dear children, it's in 3333 format, right? So the codon will be triplet. 61 codons are there that codes for amino acid, and there will be 3 codons which is not code for anything. They are stop codons. Okay, they are what, my dear children? Stop codons. So in total, how many codons do you have? 64 you have. 61 of them they are coding for amino acid, and 3 of them are not going to code for anything. They are just going to chill. Stop codon. Okay. Now AUG has dual function. One is basically to code for methionine, and the other one is that it acts as initiator codon. Now, some amino acids are coded by more than one codon, hence the code is degenerate. I told you phenylalanine is coded by UUU and UUC also. That means different codons are coding for the same amino acid. So this is degenerate feature. Now, the codon is read in mRNA in a contiguous fashion, that is without any without any punctuations, full stop, comma, anything. It's just read like that. Now, UAA, UAG, UGA, these are the 3 stop codons and they are known as terminator codons. What do you call them? Terminator codons. Now, the code is nearly universal. For example, from bacteria to humans, UUU would code for phenylalanine. I told you already. Some exceptions for this rule have been found in mitochondrial codons. In mitochondria also there are codons, okay, and in some protozoans. So, this is your answer. Please write all the points and you will get marks for it. You don't have to mug things up, you have to understand and then you have to write. Okay, cool? Chalo. Now, the last question, my dear children. Given below are the diagrams of plasmids A and B. This is plasmid A, this is plasmid B, okay? Observe meticulously and answer the questions that follows, okay? You have to observe concentratingly, okay? Now, which plasmid or is or are you select for cloning and why? Which plasmid do you think you will be selecting for cloning? I'll say that I'll select both plasmid A and B. Why, my dear children? Because over here, in the case of plasmid A, I have two antibiotic resistant genes. In one, I can put the gene of interest that I am looking forward for. Okay, for example, in the tetracycline resistant area, I am putting my gene of interest. Now, how do I find whether this has my gene of interest or not? I just treat it with tetracycline. If it is not able to survive or growth in that particular field, that means this has my gene of interest. It is not resistant to tetracycline. Okay, so that can be done. Anyways, two of them are there, so this can be used. Apart from that, if we talk about this particular plasmid, my dear children, here we have LAG-Z gene. Now, the LAG-Z gene is coding for something that is known as beta-galactosidase. It's an enzyme. And what happens when the beta-galactosidase is being released is that whenever I put some compound that is X-gal, okay, there is a thing that is known as X-gal. When I put X-gal and when the beta-galactosidase is being produced, this beta-galactosidase enzyme acts on this X-gal and produces a blue color. And produces what? A blue color. Now what I am doing, basically, I am going to insert my gene of interest over here. That means there won't be production of beta-galactosidase. Now, to understand whether this particular plasmid have my gene of interest or not, what will I do? I'll still put some X-gal and check. If the X-gal is getting converted into blue color, that means, or basically the whole colony is converting into blue color, that means beta-galactosidase is still being produced. That means it has not taken up, okay, this region has not taken up my gene of interest. But on the other hand, if there is no blue color change, that is white colonies that are being produced, that means my gene of interest has been inserted. So I can use either of them, but this will be more easy for me to understand things because there's a color difference that comes upon, right? So yes. So I can take both, but plasmid B would be recommended. Now it does not go back. Why? Yeah. Now what is insertional inactivation? I told you just right now, whatever I was talking is insertional inactivation. Whenever I am going to put my gene of interest in there, in one particular area, and that particular gene which was there, it is now inactivated because I am inserting something else over there. This is known as insertional inactivation. It is basically losing its properties, losing its property of showing the nature. Okay, so your answer becomes something like this. So both plasmids allow for cloning via insertional inactivation, but plasmid B might be preferred for the ease of screening using the blue-white method. I told you. Now, insertional inactivation is a process where insertion of foreign DNA, that is gene of interest, into the marker gene, example tetracycline in plasmid A or LAG-Z in plasmid B, disrupts its normal function. This disruption results in detectable phenotype. Okay, it will disrupt the normal function, it will make it inactive, such as loss of tetracycline resistance in plasmid A or loss of beta-galactosidase activity in plasmid B. Either way, it can happen. Okay. Now, will the number of culture plating you should make is same or different to select the recombinant if insertional inactivation is possible? Comment. So, it's a 3-marker question. How do you make them understand this? Now, my dear children, culture plating as in you want to find out whether the plasmid that is there, it has taken up the gene of interest or not. So, in the case of plasmid A, if I am doing the experiment, I need 2 culture plates. In one culture plate, I am just going to put ampicillin, okay, and see whether the things are growing or not. That's the only thing that I am going to do. Okay, I am going to put ampicillin and I am going to check the organisms which are there. The organisms which are there, they are able to grow or not. So, this plasmid is being put in the organisms. This plasmid is being put in the organisms. Suppose I am putting in this over here. So, what happens is that, okay, first thing that happens is that this is an ampicillin medium, organism that has the plasmid. Okay, I am putting my gene of interest in the tetracycline thing, but ampicillin is still there. Okay, ampicillin part is still there, my dear children. And what happened, I tried to put in both, but I don't know which of them have taken up. Now when I'm growing in ampicillin media, I will find that one of them is able to grow whereas the other is not able to grow. That means this is the one that has not taken up the thing. This is not transformed. This is a non-transformant. It has not taken up the plasmid, but this guy has taken up the plasmid. So first plating is for me to understand which is a transformant. Okay, so I got to understand that. Now what? Now I have to see whether the tetracycline gene, okay, so I am putting one more bacteria over here. So this bacteria is also there, which has the plasmid and the ampicillin resistant gene. Fine. Now I am coloring it. This is 1 and this is 2. Cool. So basically what have happened is that now I got to understand both of them, they are transformant because both of them are able to live there. They are resistant to ampicillin because they have this particular plasmid, that is why they are resistant to ampicillin. Now the second thing is that I want to find whether they are having the recombinants. Whether they are having the recombinants. That means whether in the plasmid which they have taken up, whether the plasmid has my gene of interest or not. How do I find that? Basically, I put the same thing over here. Okay. I put the same thing over here. This is 1 and this is 2. Both of them has ampicillin resistant gene. Now, let me tell you. What happens is that this one has my gene of interest, whereas this is not having the gene of interest. So now this time I am going to do a culture plating where this particular thing has tetracycline. Now listen, the one that would not be able to grow in there and the one that will be able to grow, okay, the one that will be able to grow, that means this one is a non-recombinant. Why, my dear children, why is it a non-recombinant? Because the one that has the gene of interest would not be able to grow in here. Why? Because it has lost the tetracycline resistant gene. So it won't be resistant to tetracycline. It won't be able to survive much. It won't be able to divide. So the one that is able to survive, that means it is a recombinant, sorry, non-recombinant. It is able to survive since it has not taken up the gene of interest. The one not survive, basically it is not able, it is able to divide, and this one is not able to divide. The one that is not able to divide, that means it is what? A recombinant, my dear children, because it has the gene of interest and it has lost the property of tetracycline resistance. So how many platings do you need? You need 2 platings for understanding this. How many platings? You need 2 culture platings for understanding this. Now over here in the case of plasmid B, there is only 1 plate required. Okay, where I am going to put organisms having this thing. Okay, and over here, suppose I put my gene of interest. Now I am putting X gal. Now I am putting X gal over here. Okay, if this particular thing did not have my gene of interest, the colonies that would have been produced would give a blue color. If it didn't have the gene of interest, but as of now it has the gene of interest, which is why the X gal would not have any change and it will be all white colonies that will be produced, my dear children. It will be white colonies that will be produced. Okay, and if white colonies is produced, that means basically insertional inactivation has taken place and it's working really well. Cool. So in one culture plate it can be done. So plasmid A requires 2-step plating, plate on ampicillin containing media to ensure plasmid uptake, that is transformation has taken place or not. Now replicate on tetracycline containing media to identify the recombinants which will not grow on tetracycline. The recombinants which are there, it won't go on the tetracycline medium, in the tetracycline medium, whereas the non-recombinants will definitely grow, okay, because they have the resistant gene still there. Now, plasmid B contains the LAG-Z gene which allows for insertional inactivation using the blue-white screening method. Recombinant disrupts the LAG-Z gene leading to white colonies on X-gal medium. Non-recombinants produce beta-galactosidase turning blue on the same medium. So, the one that is blue, that means they are non-recombinants, and the one that is giving you white colonies, they are the recombinants. It can be done in the same plate itself. You don't need to have different plates for that. Cool. And with this, we have completed all the questions of your model paper 1. of PU2, and I hope each and everything was really helpful for you. Do let me know if you have any queries, my dear children, and do let me know if you understood each and everything. If you did, definitely do not forget to tell me in the comment section. I know this video had been long, but I tried my best to explain you everything possible. Yeah, so that's all, and take care. Bye bye, lots of love, and subscribe to our channel.